Welcome back to my course aspects of biochemical engineering. Now, in the last lecture, I tried to discuss that how the cell concentration can be expressed, and I told you that two different type of cells are there. One is called unicellular cell, another is the multicellular cell, and or we call it filamentous type of cells. Now, in case of unicellular cell, number is proportional to mass. So, we can express the concentration of the cells both in terms of mass per unit volume or number of cell per unit volume, and all are interconvertible. But in case of this filamentous cell, or the number is not proportional to mass. So, we shall have to express only mass per unit volume. Now, then I try to discuss that how the growth model can be expressed, that that rate of growth of the cell can be expressed. So, this model can be of 4 different types. One is called segregated model, unsegregated model, structured model, and unstructured model. Now, segregated model means, as you know, that we have in our, if you look at our human population, that we will find that all the people there some way differ from each other, they are not alike. So, same thing happens to the microbial system, but all cells they grow in a different way. So, if we consider, if we put a marker to the each and every cell, and we monitor the rate of growth of the individual cells, and we write the growth equation for the individual cells, then we call it segregated model. But what is the unsegregated model? When we assume that all the growth of the all cells are uniform. And this is kind of ideal situation, not the real situation. The real situation is that the growth characteristics of the individual cells, that should be different. Now, in case of structured model, I told you the living cell comprises of different type of biomolecules like RNA, DNA, protein. Now, if your model deals with how these biomolecules changes with respect to time, then individual rate of change of RNA, how it depends on the different parameters, rate of change of protein, how it differs with the different parameters, then we call it structured model. Now, if we assume the rate of change of RNA, rate of change of DNA, rate of change of protein or other biomolecules, they are uniform, they are not same, then we call it unstructured model. So, we can easily visualize from that unstructured and unsegregated model are simplified model, what we call ideal model, and real model is about segregated and structured model. Now, Monod, similar to the Michaelis-Menten equation, they propose the equation that is mu equal to mu max S, Ks plus S. Now, this is usually this is the unstructured and unsegregated model. Now, and this kinetic constant, that mu max and Ks, we can easily determine with the help of plotting the Leimer-Bach plot. Now, then log phase, am I right? And if it is active in the log phase, our inoculation should be done in between mid log phase to the from mid log phase to late log phase. So, this I discussed in the last class. Now, today I want to share with you with other type of models that proposed by different scientists, and then we are going to discuss substrate inhibition and product inhibition, and some toxic inhibition of the microbial system, and then we shall switch over to the Luedeking-Piret and the Pirt models. Now, first let me start with that other models that are proposed by the different scientists. First model is the Blackman equation. This equation is mu g equal to mu max, and that is valid when S is greater than 2 Ks. Now, mu g is the specific growth rate, then this is equal to mu max twice Ks into S if S is less than Ks. Trizier model is mu g equal to mu max 1 minus e to the power k into s. And then that Moser model, this is mu g equal to mu max s to the power n k s plus s to the power n. Here I have seen that 1 we can find out if 1 n equal to 1, then this equation tends to monod equation. Am I right? This tends to monod equation. Now here also we have seen the Ksx we know saturation constant. Now this saturation constant is correlated with the cell mass concentration. Now if the cell mass concentration is constant, then we can assume this as a constant. Then also this will approach to the monod equation. Now as I told you that different type of inhibition that we have, that I can give you a typical example. Suppose when you plot mu versus s, this we have this kind of plot. This is no inhibition. Now in case of substrate inhibition, what will happen? This will be like this. This is with inhibition. Now why at the higher substrate concentration there will be inhibition. So this we consider as the substrate inhibition. Now during when we discuss the enzymatic reaction kinetics, we pointed out in case of enzyme we know this is a substrate. Am I right? The enzyme also we have substrate inhibition. When individual substrate they interact with the active site, they give the product. But when have two substrate try to interact the same active site, then we find some kind of inhibition take place. So this we explained during the enzymatic reaction kinetics. The inhibition pattern of the microbial growth is analogous to the enzyme inhibition. This is exactly what I want to explain. Now often underlying principle mechanisms are complicated. The kinetic constants are obtained from the experimental data per guard fitting. That we have found out. And we found out that there are three type of inhibition is substrate inhibition, product inhibition, and inhibition by the toxic compounds. So that during the enzymatic reaction kinetics, I have given one example that how the enzymatic reaction kinetics can help you to determine the concentration of the toxic component like the pesticide. So this is the similar type of things that is applicable to the microbial system also. Now first let me discuss about the substrate inhibition. At high substrate concentration, microbial growth is inhibited by the substrate, because this is the normal we have seen in our case also. We the human beings, when we start taking food, we take excess of food, our system also gets affected. So similar things happen with the microbial system also. And this inhibition may be of two types. One is called non-competitive inhibition, and another we have competitive inhibition. Now we have already seen that what do you mean by competitive inhibition and what is called non-competitive inhibition. Now what is if you have a line of a bar plot like 1 by mu versus 1 by s plot, then here there is no inhibition. This is no inhibition. Am I right? Now in case of non-competitive inhibition, this will be non-competitive inhibition. Non-competitive inhibition. Am I right? And in case of competitive inhibition, what will there? This will be competitive inhibition. This is like this. This is competitive inhibition. This we have already found in case of enzymatic reaction. Similar thing is applicable here. The equation is similar to that. Only the thing is that the different mu, the V is substituted by mu, and Km is substituted by Ks, that is the saturation constant. So this can be written, and this can be if Ki is that enzyme and inhibitor complex constant, the equilibrium constant is more than the Ks, then we can write mu equal to mu max S Ks plus S square by Ki. And competitive inhibition, we can, similar to enzymatic reaction, we can write like this. Now, substrate inhibition may be elevated by slow and intermediate addition of substrate of the growth media. What does it mean? That in the growth media, if we add substrate slowly, then this substrate inhibition can be avoided. Now, I can give a typical example, that fed batch process. What is the fed batch process? That we can, two type of feeding we can have in the reactor, either constant intermediate feeding or constant volume feeding. So, like this we can find out that your system can use, your organism can use more substrate during the fermentation process. So, this is how we can partially overcome the substrate inhibition problem. Now, product inhibition, it is similar to the substrate inhibition, that high concentration of product can inhibitory effect on the growth of the cells. We have seen that particularly in case of ethanol fermentation process, acetic acid fermentation process, we have seen that high concentration of product that have some kind of inhibitory effect. The product inhibition of growth may be competitive or non-competitive, and when underlying mechanism are known, the inhibited growth rate is approximated to exponential and linear decay expression. So, that this is like this, that that we can have three different types. Non-competitive inhibition, as we mentioned before, that this similar type of expression is there, only this is S is replaced by substrate inhibition, it was S, and it is replaced by P, and Kp is the product inhibition constant. And in case of competitive inhibition, we have similar type of expression, and in case of the other rate in linearized expression, this we can have mu equal to that this exponential type, this is mu max S 1 plus Ks by S 1 by Pm maximum product to the power n. Pm is the concentration at which the growth stops. When concentration is maximum, at the maximum concentration, growth, no growth takes place. This can be modified in this form. So, three different type of models, we have this different type of equation we can use just to describe the product inhibition. Now, I was talking about the inhibition with the respect to some toxic compounds that that may be produced during the fermentation process, or if you add some kind of toxic compounds, that that it can be expressed like this. In case of non-competitive inhibition, it will be this; in competitive inhibition, it will be this, and uncompetitive inhibition is the similar to the enzymatic reaction. All the cases, if you put I equal to 0, then all the models will be tends to the Monod equation. So, this is similar to the enzymatic reaction. I already explained, so I am not explaining again. Now that we discuss about the Monod equation for the cell growth kinetics. Now, it can be explained with the help of other equation, what you call logistic equation. It characterizes the cell growth in time of carrying capacity, that is the maximum cell mass that can be obtained. That that means this model or this equation based on that how much maximum cell can be concentration can be achieved on the basis of that, not on the basis of substrate concentration. So, basic difference between the Monod equation and the logistic equation is that Monod equation based on concentration of limiting substrate, when logistic model depends on the maximum substrate concentration. So, this expression is that the rate of cell mass formation equal to K into x 1 minus x by x. X is the concentration of cell at any time t. This is what he has written here, any time t, and xm is the maximum biomass concentration at the end, is the gram per liter, and K is the logistic rate constant. This is K is the so this can be explained like this. Now, this integral form of the above equation can be written in this form. If we solve this equation, it will come in this form. This you can write in this form. So, what is the difference between the logistic equation and the Monod equation? The Monod equation, as I pointed out, that the growth related with the biomass concentration and limiting substrate concentration. Am I right? Biomass concentration, because when we considered mu equal to what? 1 by x dx by dt. So, it depends on the concentration of the cells. And if you look at the Monod equation, mu equal to what? Mu max S Ks plus S. So, it is the limiting substrate concentration. So, it is there, and then in the logistic equation is independent of substrate concentration, it does not depend on substrate concentration, it is only related to the biomass concentration, what we have already observed, that how it is related to the biomass concentration. In the logistic equation, directly proportional to the biomass concentration and the carrying capacity, the xm minus x. Now, in case of unicellular cell, we do not have much of problem, because but we have typical type of growth characteristics in case of filamentous type of organism. As for example, Aspergillus niger. Aspergillus niger, Aspergillus armori, when you grow in the fermentation media, that when they grow in the fermentation media, what will happen? This what will happen? This you will find lot of pellet formation, the circular, the spherical pellet formation, the cells, they will form the pellet inside the reactor. So, the question comes how this we can growth model can be developed in case of filamentous organism. The filamentous organisms such as mold form the microbial pellet at the high cell density in suspension. So, this is under submerged condition, they form the pellet. Now, cell growth inside the pellet is subject to diffusion limitation. Obviously, when there is a pellet, the concentration of the cell, then diffusion of the substrate is a substrate diffusion inside the pellet is a problem. The absence of mass transfer limitation, that radius of the pellet in submerged culture increases linearly with time, as that dr by dt, dr is the change of radius of the pellet with respect we assume to be constant, the km, where r is considered as the radius of the pellet. Now, here the mass of the cell, the volume of a pellet is a we can consider this is a spherical in shape. What is the volume of the sphere? 4 by 3 pi r cubed. Am I right? Now, what is the rho? Rho, unit of rho is the volume per unit mass per unit volume, mass per unit volume. Am I right? Now, if you multiplied volume with mass, that density, so this will be obviously mass. Now, if you differentiate with respect to time, then we will get this equation, per rho for pi r square dr by dt, and we can write in this form, and finally, we can write dm by dt equal to gamma into m to the power 2 by 3, where gamma is equal to kp into 36 pi rho to the power 1 by 3. Then we can write that m is coming in this form. Now, if m we can differentiate, we will get this equation, and dm this I have already explained. Then if you go here, this is dm by dt equal to m to the power 2 by 3. Am I right? Now, if you write the m integrate, we will get this form. This is m equal to m0 to the power 1 by 3 gamma t by 3 to the power 3. Now, if we assume m0, the initial mass of the pellet is negligible as compared to final mass, then we can write this equation in this form, and this is how we can develop the correlation, and the above equation gives the cubic dependence of the mass with respect to time. So, this is how we can develop the growth model in case of filamentous type of organism. Now, as far microbial system is concerned, that up till now we talk about the rate of growth of the cells. Now, what about the product formation? The rate of how the rate of product formation is correlated with the growth of the cells. So, most of the microbial process that we grow the cell to get the desired amount of product. I mentioned the basic difference between the biochemical process and the chemical process is what? That I told you that same substrate can produce the n number of products. Here we change the type of organism in the system, because glucose can be converted to citric acid, glucose can be converted to ethanol, glucose can be converted to acetic acid. So, so question comes that how this product formation take place, how this product formation is correlated with the cell growth. Now, this has been established by the Luedeking-Piret. Luedeking-Piret proposed the equation like this, that you can see here that dp by dt, this is the equation they proposed, dp by dt equal to alpha dx by dt into beta into x, where alpha is considered as a growth associated coefficient. You see that growth associated this alpha is the growth associated coefficient, and beta is the non-growth associated coefficient. So, I can write alpha is the growth associated coefficient. Am I right? And beta is non-growth associated coefficient. Okay. So, now if you divide by x, then it will be specific product formation rate equal to alpha into specific growth rate of the cell into beta. So, obviously, we can write in the short form QP equal to alpha into mu into beta plus beta. So, this is something equal to y equal to mx plus c. This is equal to y into mx plus c. Am I right? It is a straight line equation. So, what we have seen that if this plot is this is the beta intercept is the beta value, and the slope is the alpha value. Though when in the growth model we have both alpha and beta value, then we call it mixed growth associated product. Now, in case of when beta is 0, the here beta value equal to 0. Am I right? This is passing through the origin. Then we consider this as a growth associated product. Now, in case of non-growth associated product, the beta value should not change with respect to mu. Now, you can see here that your specific rate of product formation, it should be constant. This will not change with respect to mu value. Then we call it non-growth associated product. Now, question comes that what are the different examples we have in case of growth associated product and non-growth associated product. Now, what I want to point out, suppose this is a batch fermentation process. This is the batch fermentation process, and here first we put the media, then we put the cell. Am I right? Then time to time you can draw the sample. So, if you find out that you can find out the x, you can p at different times, you can find out. This is with respect to time. You got the x p if you plot, if it is like this, and it is like this. So, this is p and this is x. Now, we should all remember the x should be it should not be 0. This is 0. Am I right? Because why it should not be 0? Because you have to use the inoculum. Am I right? The cell you are adding to the media. So, there will be some initial cell mass concentration, and from that the cell mass increase will take place. And then if your product formation, as your cells grow, if your product formation takes place simultaneously, then what we can write? dp by dt is proportional to dx by dt. Am I right? So, in that case we call it, this is growth associated product, growth associated product. Am I right? Now, if suppose when your organism attain the plateau, then your product formation start, then this is an example of non-growth associated product. Now, at that situation, dp by dt will be proportional to x. It will not be proportional to dx by dt. In case of non-growth associated product, your dp by dt will be proportional to x. Now, in case of mixed growth associated product, this is mixed growth, that what will happen, both neither it is growth associated nor it a completely growth associated product. So, both it depends on the growth of the cell and rate of growth of the cell and the cell mass concentration. Then we call it mixed growth associated. The examples are several, but in case of growth associated product, the examples I told you, there is the ethanol fermentation. Am I right? And in case of non-growth associated product, all the antibiotics like penicillin. Now, in case of mixed growth associated product is the lactic acid. So, this is the different fermentation process that we have. Now, next is the Pirt equation. The Pirt equation is a equation where when cell grow, some of the substrate used for the cell maintenance purpose. Now, what do you mean by cell maintenance? Because in a solution, if you see under the microscope, we will find cells are moving one place to others. How they are moving? They require some energy. Am I right? So, that is called cell maintenance. Not only cells, that is cells sometimes due to mechanical stirring and other purpose, cell may get ruptured, and for the recovery of the cell also they require some kind of energy, and for the formation of some protein also they require energy. So, the cell maintenance is a very important aspect that has been considered by Pirt. Now, how they have considered, what they are saying, the rate of substrate that is consumed in the system, it has dual purpose. Substrate is going for growth of the cells and substrate is going for the maintenance of the cells. Now, when you say that ds by dt, when write like the ds by dt, what I can write, this is equal to ds by dx by dt. Am I right? So, this is equal to 1 by y x by s into mu into x. So, this is exactly what he has written, mu x into y x by s overall, mu x y dash the growth plus mx. Why mx? The maintenance of the cells, it depends on the, is proportional to the concentration of the cell. More cell, more will be the maintenance, less cell, less will be maintenance. So, this m is considered as the maintenance coefficient. Okay. This is maintenance coefficient. Now, if you divide by mu x, then we have this correlation. Now, here if we plot 1 by y x by s overall and 1 by mu, then we will get a straight line. The slope will give you the value of m and intercept will give you the 1 by y dash x. That is the, this is called true yield coefficient. What is true yield coefficient? Mass of cell produce, amount of cell produce from per gram of substrate consumed when substrate is used only for the cell growth, not for other purpose. That is called true growth yield, and this is remain constant for a particular organism. Now, let us see this very interesting problem. It has been extremely argued that Luedeking-Piret model related to the cell growth and product formation and maintenance of the energy model, that is the Pirt equation, are equivalent. Do you agree? And substitute your answer. Now, what does it mean that that we by this time we know that what is Pirt equation? Pirt equation deals with the maintenance of the cells, and Luedeking-Piret model deals with the product formation. The question comes: are they equal? Are they equivalent? Not equal. Are they equivalent? Now, how we can justify that? Now, in the Pirt equation, we have this that model we have already derived, and this has come from this. Am I right? That minus ds by dt equal to mu x y dash. This is the true yield coefficient m into x. Now, y dash x by s is constant. Am I right? So, we can write ds by dt is minus ds by dt equal constant into mu into x plus constant into x. Now, when you come to the Luedeking-Piret model, we can write the substrate used for dual purpose, similar to Pirt equation. One is for growth, another is for product formation. Now, whatever substrate used for growth, I can write like this, similar to Pirt equation, and this is for from the Luedeking-Piret model. Now, if we analyze these two equation, then we can write minus ds by dt equal to this minus this, we can write this is constant and this is also constant. So, we can write ds by dt equal to constant into mu x, and this is constant into mu x, and we have found in case of Pirt model also it is same. So, from that we can conclude that both are equivalent. So, in conclusion, I want to tell that different besides Monod equation, other scientists they propose the different type of growth model for the for correlating the how the substrate concentration related with the cell mass growth, and we find that some cases we can maybe the case the saturation constant is by particular confidence model, the saturation constant is related with the cell mass growth. If cell mass growth is constant, then cell mass concentration is constant, then it approaches to the Monod equation. Then we try to explain the logistic equation and try to differentiate. Logistic equation depends on the concentration of the biomass. It doesn't related with the concentration of the substrate. And finally, I try to discuss the Luedeking-Piret model and Pirt equation, how they relate with the product formation and the maintenance of the cells. Thank you very much.